Today we are on the brink of a whole new way of treating disease, cellular therapy. In other words, harnessing the power of nature, the potential locked inside of our own cells, to repair, treat, and even cure some of the most difficult diseases of mankind. And that potential comes from stem cells. But what are stem cells? Stem cells are early-stage cells that can divide and develop into various specialized cell types in the body. They can also self-renew to produce more stem cells. In mammals, including humans, there are embryonic stem cells, and the kind we'll be focusing on for the next 3 days, adult stem cells, which act as a repair system for the body, replenishing and healing adult tissues. These adult stem cells are found in children as well as adults. They can be found in many tissues and organs, including brain, bone marrow, blood vessels, muscle, skin, teeth, heart, gut, and liver, among others. What makes these cells exciting is that they have the remarkable capacity to transform or become any number of different cell types, with some limitations. Usually they can only become cells of the same tissue type. This means that adult stem cells could be used for what's called cell-based therapies, offering the possibility of a renewable source of replacement cells and tissues to treat diseases including Alzheimer's disease, spinal cord injury, stroke, burns, heart disease, diabetes, arthritis, and more. Most of us think of a cell as a ball of liquid or gel filled with a bunch of tiny structures just floating around inside. In fact, a cell like this white blood cell is an incredibly complex and orderly organism where literally trillions of chemical reactions are happening every second. Here's a closer look. We're passing through the cell membrane, which is like a semi-fluid envelope. Those titanic icebergs floating in the membrane are sugar and protein compounds. They regulate how things like nutrients and waste pass in and out of the cell and help transmit signals to the cell's interior. Now let's go inside. The cell is held together by a complicated skeleton made up of microtubules that are constantly being built, taken down, and rebuilt. A lot more is going on here, but our destination is down there, in the nucleus. That's where we'll find DNA. As you know, our genes are elaborate combinations of nucleotide molecules that make up DNA. Some genes help cells become bone, others to become skin, muscle, or liver cells, and so on. This is what's called differentiation. In an embryonic stem cell, virtually all the genes are open for business, meaning the stem cell both contains all of the genetic code needed to turn it into every type of cell in the human body, and it's all available for use. But as an embryonic cell starts to differentiate, as it starts to become a specific type of cell, unneeded DNA shuts down. Inactive genetic material, or heterochromatin, is formed. And while the unneeded genes are still present, they're just not accessible. We used to think that was the end of the story, that adult cells were fully and irreversibly differentiated and only capable of making more copies of themselves. But now we know better. Turns out we keep some stem cells around. Technically, they are progenitor cells, meaning that while some of their genes have shut down, not all have. So they can be reprogrammed or reactivated to a certain extent to make more of the tissue they're found in. For our purposes, we'll call them adult stem cells. And we're now learning that these adult stem cells are found in many, perhaps most, tissues in the body. These cells are said to be pluripotent. The example here is bone marrow stem cells that may be able to differentiate or mature into liver, lung, GI tract, and skin. It's thought that these pluripotent cells are the cells we call V cells, short for very small embryonic-like stem cells. But now scientists have figured out an even neater trick: how to take fully differentiated cells and reprogram them into stem cells. It's done by taking the shut down genes and opening them up for business again, either unwinding the tightly wound inactive DNA or by inserting some key genes the cell needs to revert to its embryonic-like state. This is usually done using viruses with the needed genes engineered into them. The virus then does what it often does best: inserts its genetic material into the human cell's DNA. And when these cells are cultured, some of them actually de-differentiate. Not only do they look like embryonic cells, they have the potential to become many different tissues. What's particularly fascinating about stem cells is that they transform by being put together with the cells we want them to become. It's a sort of a cellular version of, when in Rome, do as the Romans do. As you've already heard, contrary to previous belief, adult stem cells can be found in many tissues, ranging from fat, heart, breast, GI tract, to even teeth, hair follicles, and brain. But by far the richest and most varied source of adult stem cells is found in bone marrow, so let's start with that. During an orthopedic procedure, for example, the bone of the pelvis is punctured and bone marrow extracted. The marrow is washed and spun in a centrifuge right in the operating room to separate out the stem cells. The cells are then injected into the patient's hip. This procedure has been used to revitalize a dying hip, a condition called avascular necrosis, and also to grow new bone to knit fractures that won't heal. But there's two drawbacks to this approach. It's invasive, and it depends upon having a source of healthy stem cells at the time when there is already disease or injury present. The much preferred technique is to bank stem cells well before a need arises. To do this, bone marrow is stimulated with a natural growth hormone. As a result, more stem cells are produced and released into the circulating blood. After a couple of days of hormone stimulation, the patient, or more appropriately the healthy donor, is placed on a pheresis machine like this one. Blood is taken from a vein in the arm, washed and spun to concentrate and separate the stem cells. The remaining blood is returned to the donor via a vein in the other arm. In some cases, these cells can be grown, we call it expanded, in a lab to increase their number. They can then be used right away or kept in tissue culture for some time, waiting to be used. Here, lab-expanded stem cells are being injected into an arthritic knee to regenerate cartilage. The drawback to the tissue culture is that it can't be stored for very long. However, there is an alternative. Stem cells can now be frozen and stored for use years, even decades into the future. This process is similar to cryopreservation or freezing of other tissues. After being typed, the cells are protected and cooled in a very precise way to avoid forming damaging ice crystals. The controlled cooling continues until reaching the temperature of liquid nitrogen. At this point, the cells are in suspended animation, virtually immortal for as long as they are kept in liquid nitrogen. When the need arises, the cells can be carefully thawed. The preserving fluid is washed out and the cells are ready for use, either for a full marrow transplant, disease treatment, or other tissue regeneration.